All right, good morning, class, or good afternoon, or good evening. This lecture is going to be on Chapter 1, A View of Life. Chapter 1, A View of Life. So how do we define life? You should know what biology is because you are taking AP Biology, right? I'm hoping that is true. So biology is the scientific study of life. There is a great diversity amongst living organisms. Living things are composed of the same chemical elements as non-living things, and they obey the same physical and chemical laws that govern everything in the universe. So in this course, we are going to study all living things as they pertain to life. Now, life is organized. So taxonomy is the branch of biology that identifies names and it classifies organisms. So classification is done in specific categories. Now let's look here. Here's our kingdom, phylum, class, order, family, genus, species. What do you notice as we go from the top to the bottom? You should notice that the organisms become less. And why do they become less? Is because we become more specific. This larger category, kingdom, contains all of the animals, hence it's in the kingdom Animalia. So when we look at here, we have bears, black bear, grizzly bear, giant pandas, red fox, Albert squirrels, coral snakes, and then sea star. As we start going down each level, we start losing an organism. And we lose an organism because these organisms here are becoming more specific or specified. The phylum Chordata contains organisms that have backbones. As we can see, the starfish doesn't have that, so it would not be listed in this phylum. Next, we get into the class Mammalia, or these are mammals. Snake is not a mammal, so that would be separated into a separate category. We have the order Carnivora, and carnivore is going to include all of our carnivores. So now we got rid of the squirrel. Then we have our Ursidae, or our bear kingdom. Now we're getting more specific. So we have our genus, which is Ursa, until we finally get the main species, Ursus arctos. Okay. Life is then classified into three large, huge domains. So these are the domains of life. Now, when I was in school, there weren't domains, we were at kingdoms. So domains are fairly new, but not. So we have our first domain, which is called archaea. And archaea contains unicellular prokaryotes that live in extreme environments. So these are bacteria. Prokaryotes, remember, they lack a membrane-bound nucleus. Okay? Where would we find archaea? These are bacteria that live in heat-loving areas, such as deep ocean vents or thermal vents. Okay? Then we have the domain bacteria. And our domain bacteria contains unicellular prokaryotes, or bacteria, that live in all environments. They live inside of us, they live outside of us, they live everywhere. Then we get into our specified domain called eukaryote. And if you look at the term eukaryote, that means that these organisms are eukaryotic cells, right? They contain unicellular and multicellular eukaryotic cells, okay? Eukaryotes contain our membrane-bound organelles. All right, like we said with our kingdoms, domain archaeas and the domain bacteria, they don't have kingdoms associated with them. Okay, why? Because these are two specific different types of bacteria. However, our domain eukaryote, they do have specific kingdoms. They have the kingdom protists, they have the kingdom fungi or fungus, our plant, and then we also have our animal kingdom. All right, here's a video for you guys to watch. It is on classification, so you could do this on your leisure. It'll give you a good idea of how we classify organisms. All right, in this upcoming weeks, this is what we're gonna focus on, is basically the process of science. You guys will be doing science, even though you are at home. I am gonna have you guys practice doing science in your households. All right, so let's look at the scientific method. Obviously, as we start with, it's a standard series of steps used in gaining new knowledge through research. Okay, notice the term research. Whenever you research something, what are you doing? Right, you're basically looking at background. Okay, you're looking through the internet. You're actually questioning what's going on. Okay, the scientific method contains a whole series of steps. As scientists, we make observations and our inferences or guesses. we ask questions, we develop hypotheses, we design experiments, we make measurements, we collect data, we interpret data, we draw conclusions, and then we communicate. So our first step is observations. As scientists, we use our senses to gather information about a phenomena or a natural event. So some of the senses that we use would be like our sense of smell, our taste, our touch, our hearing, as well as our sight. So we see things, we hear things, we feel things. Okay? And then from there, we form what's known as a hypothesis. So a hypothesis is a possible explanation for a set of observations or answer to a specific question. Okay, it can be something that can be tested, and we word it as an if-then statement. Hypothesis can either be supported or disproved. So an example, if a superball is dropped from increasing heights, then the bounce heights will also increase because. This because is what you think would occur. Okay, so once again, our hypothesis is a possible explanation for a set of observations or answer to a scientific question. And we are going to practice doing a whole ton of hypotheses this year. All right, so what I want you guys to do is go ahead and take a look at this photo. And I want you to form a hypothesis in your head using the picture below. Notice what you're seeing. Look at the background. Look at the weather. Look at the ground. And obviously look at the trees. What do you think is occurring? I'm going to give you a few seconds. I want you to think. This photo was actually taken last week. I went to Yellowstone for the first time and I visited Yellowstone National Park. And these trees were located where the thermal vents are located. As you can see, steam kind of flowing up in this area. It's not clouds, it's like steam. Here are clouds. See the difference? Okay. Now, these areas are filled with water that is highly acidic. So as we can see here, the acids have burned through the leaves. These trees used to look like this. Okay. So my hypothesis would My hypothesis would state that the trees living in environments that are acidic will not be allowed to grow. Okay. We can also state it obviously a little better. If trees are planted in an area of high acidity, then they will not grow. All right, let's go on. In biology, we also come up with what is known as a null hypothesis. So that's something new that you learn in this class, a null hypothesis. And it's kind of a terminology that we use with statistics. So a null hypothesis is basically a hypothesis that can be rejected. It essentially states that the variables will have no effect on the outcome of the experiment. So then why do we use a null hypothesis? Because you can never have enough support for hypothesis to say that it is the sole explanation of a phenomenon, but you can have enough data to show that is not the sole explanation of the phenomenon. Oh, great. Okay, what does that mean? All right, let's go back to our photo here. So as we said if trees are planted in an acidic environment, then they will not grow. So how would I write a null hypothesis? So in a null hypothesis, I would state that plants grown in an acidic environment trees grown in an acidic environment will have no effect on the growth. So trees grown in an acidic environment will have no effect on their growth. Well, I know that would not be the case because I know that trees can't grow in that area, okay? So a null hypothesis, once again, is a hypothesis that can be rejected, and it states that the variables have no effect on the outcome of the experiment. So what I'm saying is acid has no effect on plant growth, that all trees would be able to grow on acidic environments, and I know that's not the case. Okay, so that's a null hypothesis. Here's another example. So the null hypothesis assumes that there's no relationship between two variables and that the controlling one variable has no effect on the other. So let's look at this one. Cats show no preference for food based on shape. Okay, so if I look at this cat, is it going to like the food here that look like fish or the food that look like triangles? Okay, if we see in the experiment that the cat is only preferring this type of food based on the shape, then my null hypothesis obviously would be invalid. Okay, let's look at this next one. Plant growth is not affected by light color. So we're saying that all light has the same effect on plants. Okay, notice the picture. We see that this plant here is obviously growing towards this light color. Here's another null hypothesis. Age has no effect on music ability. Age has no effect on music ability. Okay. We would hopefully think that as you get older and wiser, if you are a violinist like this lady here, she'll probably have a little bit more experience. So you would think that her musical ability would be much greater than that of the younger girl as we see in this photo. All right, if you get stuck, here's another video on what a null hypothesis is. And next week, we will be doing some worksheets in which we are going to be practicing the null hypothesis. All right, once we have our hypothesis, we then can design a controlled experiment. A controlled experiment is an experiment in which all of the variables except for one they remain the same. Okay, an experiment that is designed to compare an experimental group with a control group. So ideally, if you recall, a control group and experimental group differ in only one variable. Whenever we're doing an experiment, we only change one thing at one time and we leave everything else the same. Our control group serves as a comparison to measure against. So our control group is that which we are measuring against. Our variable, our experimental group, is that what we change. So let's look at this. A variable is a factor that can change in the experiment. So what exactly are you changing? We have two variables which we look at. We look at the independent and the dependent variable. The independent variable is your manipulated variable, and that's the variable that is changed during the experiment, okay? So if I was looking at my plant growth, right? One thing that I can change in which plants grow would be light, or the amount of light or the color of light. Now, if I change the color of the light, how am I going to measure whether the color of the light affected my plant growth? That would be the responding or the dependent variable. That's the factor that changes because of the manipulated variable, or that's what's being measured. So I have the thing that I change and how I measure it. If I change the light, my measurement then would be my growth of the plant. So the growth is the dependent variable, and the change of light is going to be my independent variable. Now look at the picture to the right. I want you to name the independent variable and the dependent variable, as well as the controlled variable. So I have the one thing you change, so it's limited to only one in the experiment. My dependent is the change that happens because of the independent variable. And my controlled is everything I want to stay the same. So my independent variable in this case is going to be the liquid used to water the plants, okay? My dependent variable, what I am measuring, is going to be the height. Notice that I'm changing the height in all these, or I'm actually measuring the height. And my controlled is the things that stay the same. So what I'm using the same is going to be the same pot, the same amount of liquid, and the same soil, as well as the same plant. Our constants, okay? Constants are separate than your controlled. Okay, so constants are things that stay the same. It's also known the controlled variable. So let's look at this example here. So here's my independent variable. So let's say, for example, I'm doing an experiment in which I'm adding fertilizer, okay? Now, what I'm measuring can either be the plant growth or the number of leaves or the number of fruits in this experiment. So that's how I'm measuring whether fertilizer works. Now, what I'm going to keep the same throughout the experiment is going to be, even though these plants here have fertilizer, in order for all plants to grow, they still need water. So water would be a controlled variable. the amount of other fertilizers, and then obviously the amount of sunlight. So things that stay the same are our controlled variables, also known as our constants. So as we said, the constant is a variable that does not change for the duration of an experiment, and it's a value that remains the same. Here's a brief video on scientific variables to help you guys understand the difference between the control group, the independent, and the dependent variables. All right, once we have that, now we gather our data and we want to interpret that, right? So you can do the experiment using the method you came up with, and then you record the results. After the data is collected, you must be organized, and then that group needs to be interpreted. So you can either create data tables, graphs to represent the data. You then perform calculations that will determine what the results mean in terms of averages, percentages, and totals. And then you can possibly identify any source of error. So we always account for sources of error in biology. Why? Because the sources of error can have some sort of effect in terms of our data collection. So we always list them as a source of error. What are some things that may have gone wrong? What are some things that may have possibly gone wrong? Because let me tell you, no experiment is perfect, no matter how many times we try and replicate it. Here's some example of data collections. In this area here, we're looking at a graph, obviously, and this one we're looking at a data table. So this is different ways in which we could collect and then analyze our data. Finally, we conclude. This is the most important part in my class, and basically in all of your science classes, is your conclusion. So this here is where your data is analyzed and interpreted to determine whether the hypothesis is supported or not. So if your prediction occurs and your data is correct and it's in line with your hypothesis, then your hypothesis would be supported. Okay? If your data is not correct and it's completely opposite from your hypothesis, that means your hypothesis is rejected. Now, even though your hypothesis is rejected, that does not mean that you did the experiment wrong. That just means that your data is different than what you projected. Okay? And if that's the case, then you can recommend a revision of your experiment. Okay? From then, our findings are reported in scientific journals. Our peers review them, and then other scientists then attempt to duplicate or dismiss the published findings. Okay? So once again, a review, we have our observations, hypothesis, experiment observations, our conclusions, and then our scientific theory. Okay? Once again, once we have completed our experiment and rerun it again over and over and over again, once we start getting the same data, then your experiment can become a theory. And these are concepts that join together two or more well-supported and related hypotheses. It's supported by a broad range of observations, experiments, and data. Okay? Now, scientific principles and laws. There's a difference between a theory and a law. Theories, in essence, can still be disproven. Most likely, they're unlikely to be disproven, but they still can because there's still a small amount of uncertainty. Whereas a law is an accepted set of theories, and there's no serious challenges to the validity. So law is almost like a fact, okay? Fact versus a little bit of uncertainty here. I want you guys to actually watch this video on theory versus law. Gives you a very good perspective of the difference between the two. Here's some basic theories of biology that you guys are probably familiar with. We have the cell theory, homeostasis, gene theory, ecosystem theories, and then our theory of evolution. Here's some examples of how we would collect our data and run our experiment. So we're looking at pigeon pea and our winter grass in terms of our experiments. We have our control group. We have our test pots. Notice what we're testing that is different nitrogen levels, okay? And then we are looking at our growth of our plants. Here's another example of a field study. So whenever we are doing a field study is when you're actually observing data. So you're not getting a numerical value in terms of like measurements, but you're actually looking at maybe organisms in a specific environment. Okay, so if that's the case, then this would be more of an observational data. And we could still collect our numbers, our numerical values as we see here, and then also interpret them into a graph. All right, our second part is the diversity of life. So in the diversity of life, despite diversity, all living things share the same basic characteristics. Now, we jumped into the next section. So like I said, in the first couple of weeks, we're going to talk about the scientific method and we're going to apply it to everyday life. And now we want to go ahead and focus on the second part, which is our characteristics of living organisms. So what do all things have in common that characterize them as being alive? Okay. So some things that all living things have in common is that they are organized. So the level of biological organization ranges from atoms to biosphere. And we know that the cell is the basic unit of structure and function in all living things. We have unicellular or we have multicellular organisms. And each level of organization is more complex than the level preceding it. So as biological complexity increases, each level acquires new emergent properties. And as we see here, we go from the least to the greatest. And all of these have an effect on each other. In this class, we're going to see how this affects this and how these small things go up. Okay, so we have the biosphere, which is made up of the ecosystem, which is made up of our community, which forms our populations, which form our organisms. Organisms are made of organ systems. Organ systems are made up of organs. Organs are made up of tissues. Tissues are made up of cells. Cells are made up of molecules. And molecules are made up of atoms. So I'm going to give you a big example. Let's say there's something wrong with the chloroplast. Okay, so I want you to visualize a plant. What would happen if there was something wrong with the chloroplast? Let's say the chloroplast had some sort of mutation. So think in your head. If you recall, what does a chloroplast do? Okay, you're right. A chloroplast is where photosynthesis takes place. And if you recall what photosynthesis is, take a second. What does photosynthesis? All right, photosynthesis is where the plant takes in sunlight and it makes carbohydrates. And that occurs at the chloroplast. Now, if my chloroplast is not functioning, that means that my plant cannot make a carbohydrate. And if you recall, a carbohydrate is a fancy name for a sugar or glucose. Plant can't make sugar. What's going to happen to the organisms in our community? So let's think about that community. Notice in this area I have elephants and I also have zebras. Well, if my plants can't make sugar, right, then that means that it's probably not going to grow properly. If the plant doesn't grow properly, that means that it's going to stunt the growth of the tree. It stunts the growth of the tree. How is that going to affect the zebras and the elephants? You're right. Well, zebras and elephants obviously are herbivores and they do eat leaves. They eat certain types of leaves from the trees. Now, if there's something wrong with this chloroplast, this population would decrease. If this population decreases, it would then affect the zebras and the elephants as these two organisms eat these types of leaves. Okay, and as we could see, it would then affect the greater ecosystem as we know it. So even though something small happens here, it can highly affect the areas located up here. All right, another property of life is all living things acquire materials and energy. So energy is the ability to do work. You and I, in order to function, need a lot of energy. Where do we get our energy from? Okay, ultimately, it comes from the sun, you're correct, but we get all our energy by the foods that we eat. So energy is required to maintain organization and conduct life-sustaining processes such as chemical reactions. Metabolism is all of the chemical reactions that occur in a cell. You need to know this term. In this class, you have to know it. So metabolism is all the chemical reactions that occur in the cell. Now, if I look at you guys, which are teenagers, your metabolic reaction rate is extremely high. Why is that? Because you're a teenager, your cells are still programmed to grow and divide at a much quicker rate than mine is. So younger individuals such as yourself has high metabolic rate. That means that you can eat food and your body's going to break it down a lot quicker than somebody who's a little bit older. The sun is the ultimate source of energy for nearly all life on Earth. We know that plants, algae, and some other organisms capture solar energy and perform photosynthesis. And if you recall, like I said, photosynthesis is the process that converts solar energy into the chemical energy of carbohydrates. Here are some organisms acquiring nutrients and energy as we see here. All right, another thing that considers an organism as being alive is all living things must maintain homeostasis. So this is another term, highlight it, know this term. Homeostasis is the maintenance of internal conditions with certain boundaries. It's the ability to maintain a state of biological balance. We have feedback systems which monitor internal conditions and make adjustments. So let's look at here at this person. This person here is obviously, hopefully you can see that they are jogging. If you are exercising, exercise affects your body temperature, your water amounts, as well as your blood sugar levels. Why is that? As you are running, your body temperature increases. As it increases, your body has to release that energy in the form of sweat. Sweat helps us to cool down, okay? In order for our cells to get us to continuously function, we need an ample amount of sugar or glucose levels. So we have our pancreas, which is going to regulate the amount of sugar. It will then release that sugar into our muscles, which is going to help us to function and run, walk, and do what we're doing. Okay, our kidneys are going to help us to maintain that water balance so that we're not losing too much water by sweating. Okay, if our kidneys fail to function, our bodies probably will not be allowed to sweat, and then we can overheat, and overheating can cause death. Okay. All right, living things also respond to stimuli. Living things interact with environment and respond to changes in the environment. The ability to respond often produces movement. Notice as we see in this person's hand, he goes to grab the pan that is hot and obviously lets go. That's a reaction. This is an extremely important reaction for us to respond to our environments. Notice this picture here, these newly grown plants, or where do we see that they're growing? Exactly, to the light. If I were to move this plant around, these plant species here are going to grow the opposite way. So these are always going to grow towards the plant, Living things reproduce and develop. So all living organisms must reproduce to maintain a population. The manner of reproduction varies among different organisms. When organisms reproduce, they pass on copies of their genetic information in the form of genes to the next generation. Genes determine the characteristics of an organism, and genes are composed of DNA, also known as deoxyribonucleic acid. So I posted this picture here because one, obviously I love Dobermans, but who doesn't love puppies, right? So as we see here, dogs have more than one organism. Their bodies are enabled for them to have more than one dog or one puppy. Why is that? Because these organisms here, if you've known, all dogs can have obviously multiple births, but we do know that sometimes some of these puppies don't survive, and they may be too small, they may be born with a mutation and they die. So dogs produce more than, they overproduce to ensure that some of these puppies will survive, okay? All right, living things have adaptations as well. So an adaptation is any modification that makes an organism better able to function in a particular environment. The diversity of life exists because over long periods of time, organisms respond to changing environments by developing new adaptations. So what do we have as humans that allow us to adapt to our environments? I want you to think real close. What does a human have that allows us to adapt better in our environment than some other organisms, like some other animals? So one thing that we have is we have opposable thumbs. So if you go ahead and look at your thumb, what do you think the purpose of your hand and your thumb is? Okay, you're right. It is used to grab and turn and twist things, okay? Humans also have our brain adaptations. We are the only organisms that study ourselves. So listen to that. We are the only organisms that study ourselves, okay? Because of that, we can improve on our species, which is actually pretty cool. Evolution is the change in the population of organisms over time to become more suited to their environment. Here's obviously a picture of a cheetah, and notice the different adaptations that allow this cheetah to survive in its environment, okay? It has a spotted coat for camouflage. It has a tail, which is used to improve balance. It has muscles or heavily musculated limbs to help the leopard pounce on the prey. It has powerful jaw muscles to help devour the flesh. And it has expendable claws on each toe that help the leopard to hunt animals and to climb on trees. It's pretty cool. All right, that will do it for our lecture. I know that was pretty lengthy, and I'm glad you guys are still with me. If not, you could have paused this lecture during any time. So I hope you learned something new. You should have learned about the scientific method as well as the characteristic of all living organisms.